It is my pleasure to introduce to you Professor Pauline Rudd. She has a bachelor's degree in chemistry from the University of London, and then she got a PhD in glycobiology at the Open University of the UK. I've been lucky enough to talk a lot with Professor Rudd during the weekend, and I found out that she had 4 children when she got her PhD. So she's also, and also she has faced several difficulties when being a woman and getting in a leadership position. So that increases even more the admiration I already had from her. So also she's a very international person. She has been a visiting professor or researcher at the US, in Israel, New Zealand. She just told me in Cuba. She has also spent a lot of time. She also has a joint appointment with in Singapore, so she spends a lot of time there. So that's also something to admire. But currently, she's a professor and principal investigator at the National Institute for Bioprocessing Research and Training in Ireland, and she's leading the glycobiology group. Thank you very much for accepting our invitation and being here for me. Thank you.
Thank you very much, Laura. Thank you for inviting me here and giving me such a fantastic weekend when I started at last to learn how impressive the archaeology is in Mexico. My youngest son is an archaeologist, and he's been trying to persuade me to visit Mexico for a very long time, and I see now I should have listened to him before. So thank you very much for your lecture, which was very helpful to me because a lot of the things you said I'm going to build on. So thank you for your introduction. So as Laura said, I come from the Irish National Institute for Bioprocessing Research and Training. So this is our building. This is an organisation that was created by the Irish government maybe about 10 years ago. And it was very far-sighted because Ireland, like Mexico, is striving to become a manufacturer of biotherapeutics and would also like to do more than just manufacture. We would also like to be able to do some research and particularly be considered experts in analytics. So this is the building that we have. We do a huge amount of training. We aim to train operators as well as master students. The idea is that a lot of people in Ireland are already making small molecules, but they need to be upskilled in order to be able to make the protein drugs which are now coming on stream. So we have the most amazing facilities for training. You can come and you can use all this equipment, which is exactly as you would find in a pharmaceutical company. And you do it all under GMP conditions. So people really learn skills which are important to the industry in Ireland. So we have upstream and downstream processing, and we have fill-finish operations, and we have analytics. So to talk about glycoproteins, well, at least 60% of natural proteins are glycosylated. And these glycoproteins can be secreted, like the ones here, IgE, IgG, and erythropoietin. Or they can be discharged in granules, like gelatinase B. They can be shed from cell membranes, like MUC1. And they can be attached to cell surfaces, like the prion protein, the influenza surface glycoprotein, HIV surface envelope glycoprotein, and the Fc gamma receptors. So the chances are, if you're working in biochemistry, sooner or later you will be dealing with a glycoprotein, because around every cell is this glycocalyx. Everything in black here is a glycan. So you can see if you're going to deal with cells, you have really to understand the importance of the sugars that are attached to the proteins and the lipids on the cell surfaces. So we all know the fundamental building blocks of nature. We know about chromosomes and the genes, and we know that they have made up of bases. DNA is made up of 4 bases, and we know that these code for gene products, for RNA to start with, but then for the polypeptides, for example, erythropoietin. And polypeptides are made up of more or less 21 amino acids. And like DNA, they're simply linear sequences. They may be folded into complex shapes, but actually they're linear sequences, which makes them pretty easy to analyze. Attached to the protein are the sugars, and the difference between analyzing the sugars and everything else is that the sugars have these complex branch structures. So you can't just start at one end and sequence to the other. You have to be more clever than that in order to determine the structure of these branches. And together, all these systems produce glycoproteins such as erythropoietin, which, as I'm sure you know, is used to treat anemia in cancer. So it is a very important glycotherapeutic. Now, glycosylation takes place in the secretory pathway. So cells which express proteins which are destined to have sugars attached to them have a leader sequence on the front end of the protein, which enables them to enter the endoplasmic reticulum through this pore called Sec61. So the newly made amino acid chain goes through Sec61 into the endoplasmic reticulum. And then, as you heard earlier, it collects a big precursor set of glycans, and the protein here is, at the same time as doing some glycan processing, it's also folding and undergoing quality control, and anything which is misfolded is being sent out into the degradation pathways. And the sugars play an important role in all of these functions. But once the protein's properly folded, it then exits into the Golgi apparatus. And this is where you have the elaboration of the N-linked glycans and the attachment, and then the elaboration of the O-linked glycans. So sugars are attached co- and post-translationally to the protein. So this is the primary amino acid sequence. This sequence here, as you heard, is the key for the attachment of N-linked glycans to the asparagine residue. So wherever you see asparagine, some other amino acid, either serine or threonine, then you can have an N-linked glycan attached. And here's IgE. These are all N-linked glycans attached to IgE. Here's PSA, has a prostate-specific antigen, has one N-linked glycan attached. The prion protein has two N-linked glycans, and this is showing you the dynamics. So these glycans are not static, they're actually moving all the time. So you can see how much of the protein surface is covered by these huge sugars. And then O-linked glycans are attached to either serine or threonine. And often these serine-threonine residues are in long domains. They're serine, threonine, proline-rich domains. That is a code for the protein to attach O-linked glycans in the way that MUC1 does. So one of the really important things to understand about glycoproteins is this idea of glycoforms. So one protein backbone can have many different sugars attached to it, and each site can have a whole set of sugars attached to it. And this is because of the processing pathway. So this is the glycosylation sequon. Here is the oligosaccharide transferase enzyme, which is going to put this big block of sugars onto the nitrogen in the asparagine side chain of the asparagine residue. And then these enzymes are going to cleave back these glucose residues, and then they start to cleave back the mannose residues, and eventually the properly folded protein goes into the Golgi, and then, as you heard earlier, these other enzymes all build up the branches to make these complex biantennary glycans. And sometimes they can be triantennary or tetraantennary. And so you can see each of these steps is carried out by an enzyme. Now, you know that mostly enzyme reactions don't go to completion because they're to some extent reversible, and also in the case of glycans, the glycoprotein is moving through the system all the time. So not every copy of the glycoprotein will see every enzyme. So sometimes IgG will come out with a fucose residue in the core, and sometimes it will miss the fucosyl transferase step and it will come out without a fucose. So you can see, if you think about that with all these different enzymes, it's possible to get many structures at one glycosylation site. So added to the complexity of the branch structures, this is another thing that makes glycan analysis far more challenging than looking at either proteins or DNA. We have, when we analyse glycans, to specify a number of different things. We have to understand the monosaccharide sequence. So we may be able to do a monosaccharide composition analysis and we'll know which sugars are there, but how are they linked together? How do we know the order that they're linked together? And each monosaccharide can be alpha or beta linked to the next one. So they can be linked above the plane of the ring, or they can be linked below the plane of the ring. So you have to specify whether the linkage is alpha or beta. And then you can have one sugar attached to another sugar at many different positions. So it can be attached at the 2, the 3, the 4, or the 6 position. So you have to tell someone, I've got an alpha-linked sugar, and it's attached at position number 4 or position number 3. You have to define this. And, of course, the branching occurs because you can have more than one monosaccharide attached to another. So here you can see there's just one attached, but you could have another one attached here. So you have also to be able to define this. So this is a very simple sugar, just a biantennary sugar with 2 N-acetylglucosamine residues attached to this trimanosyl core. And you can see here that once you start to look at all the linkages and all the anomoricity, it's actually quite complicated. And in fact, this is the UPAC formula for this sugar, which we represent as this simple symbol. And you may say, well, really, does it really matter? Well, it matters a great deal because monosaccharide units can provide recognition motifs. So this is a simple mannose monosaccharide, and this is a simple glucose monosaccharide, and a simple galactose monosaccharide. And you look at it and you think, well, it's just a sort of mess of hydroxyl groups and hydrogen atoms. Why does it really matter where these hydroxyl groups and hydrogen groups are attached? Well, if you look in a bit more detail, you can see in mannose and glucose, see these hydroxyls are actually cis to each other. They're in the same orientation. But if you look at galactose, you've got a hydroxyl group here and a hydrogen atom here and a hydroxyl group here. So these are trans hydroxyls because the hydroxyl groups are trans to each other, not cis. Now that actually makes a lot of difference to how these glycans can interact with proteins, because there are a lot of proteins which are called C-type lectins. And these C-type lectins, so this is one, the mannose binding lectin. It has 3 carbohydrate recognition domains, and this is one of them. And inside these domains, there are calcium ions. And the calcium ions can ligate to hydroxyl groups. But the ligation, the calcium has very strict control over the geometry of the ligation positions. So it can't just bind any hydroxyl groups. The hydroxyl groups have to be oriented in such a way that they can actually bind to the ligation positions in calcium. So here's the mannose binding lectin protein. These are the amino acid residues that hold the calcium ion in place. And then you can see some of the ligation positions are taken up by the protein. But these 2 here are free. And if you can present it with sugars which have hydroxyl groups in just the right position, then you can ligate this sugar to the mannose binding lectin. And the only sugars that have hydroxyl groups in the right positions are mannose, glucose, N-acetylglucosamine, and fucose. Now, why am I telling you all this? Because if you're using IgG for therapeutic purposes, a lot of IgG ends in N-acetylglucosamine. N-acetylglucosamine has these hydroxyl groups pointing in these directions and can ligate to the calcium. So it can actually bind to the mannose binding lectin. If your sugars end in sialic acid, then sialic acid can't bind to the mannose binding lectin, and you won't invoke any function that involves it. When it comes to O-link glycans, their processing is completely different. So it starts in the Golgi, the protein is already fully folded, and you're going to be adding an N-acetyl-galactosamine to the hydroxyl group of either serine or threonine. Now that serine and threonine has to be able to present this hydroxyl group so that it can be seen. So if it's buried deep in the protein, that site will never have an O-link glycan on it. So if the hydroxyl group is exposed, then this N-acetyl-galactosamine residue gets attached, and then it becomes extended, one monosaccharide at a time. And there are a number of different core structures for O-link glycans, which you can see some of them here. So they all have GalNAc residues attached to the protein, but then they have different monosaccharides attached to the N-acetyl-galactosamine. And here you can see a bit more detail. These are some of the common core structures. So 1, 2, core 3 type 1 and core 4. And these are the mass spec profiles showing the fragmentation. So if you want to be able to identify these glycans, you need mass spectrometry, because often the same residues are present, but they're in different linkages. And looking at the way that these molecules break up tells you what the different linkages are between the different monosaccharide groups. Now, O-link glycans are important to erythropoietin, actually, because erythropoietin has an O-link glycosylation site just here. And in fact, it was the subject of, there was a huge patent dispute about 15 years ago, and a lot of it was around which sugars were present at this O-link glycosylation site. But erythropoietin has these 3 very large sugars, and remember, they're not static, they're actually moving. So these massive sugars attached to this relatively small protein. And the protein has to interact with the erythropoietin receptor, which you can see here in white. It's a dimer, and it comes, it's not a strand, but 2 molecules of the receptor link to each molecule of erythropoietin. So if this reaction is going to happen, it's really important the sugars are not in the way. So sugars are never in the way of the primary function of the protein, because otherwise there'd be no point, the protein wouldn't work. So you can see here, and in this sideways view, you can see how the sugars are all somewhere other than blocking these interaction sites. And so I'd just like to just run through this very quickly, just to say that now on I'm going to be using these symbols for the different monosaccharides. So the model that we have is that hexoses are open symbols, like glucose, galactose, mannose, and then hexnacs are, glucnac is a glucose shape, but it's filled in. And this is a galactose shape, but it's filled in. This is deoxygalactose, so it's got a dot in the middle. And we show the linkage positions by the way that we join these monosaccharide shapes together. So this, you can see this is in a fucose, because it's that shape, and it's in a 6th position, because it's linked in this direction. And also, if you want to know whether it's alpha or beta, you can see it's alpha, because it's got a dotted line. So these cartoons actually give you a lot of information. Now, there are many glycoproteins in the biologics market, ranging all the way from blood proteins to cytokines to monoclonal antibodies and bone proteins. So a whole range of them are now validated by the FDA. And they range from interferon, IgY, this is a chicken immunoglobulin, erythropoietin, tissue plasminogen activator, which was one of the very first biotherapeutics. And this is a monoclonal antibody again, and etanercept, which is also a monoclonal antibody. So most of these, in most of these glycoproteins, the sugars are really important for the function, which is why everyone now wants to understand the function and the structure of the sugars on their therapeutics so that they have safe and efficacious drugs. So just a few roles for glycans, just to run through. Protein folding. Entering calnexin and calreticulin pathway, that depends on glycosylation. Also, it affects the local protein secondary structure. If you've got a big sugar attached, it's not going to fold in on itself. It's going to, the sugars are going to be exposed on the surface of the protein. So if the sugar's not there, you can really affect the folding, and you can promote aggregation. Protein targeting, trafficking, sorting, directing a protein to its final destination. Sugars are also important for ligand recognition, which I showed you with the mannose binding lectin just now. They can either bind themselves to receptors or they can modulate receptor binding. They're important for the biological activity. For example, gonadotrophin signal transduction doesn't happen unless the sugars are attached. They're very important for stability. The sugars decrease the rate at which the protein breathes. They enhance the solubility. They shield hydrophobic patches. They protect the protein from proteolysis. They're involved in immune recognition, and they stabilize intra-chain interactions. They also regulate protein half-life. So if you have sialic acid, you can increase the half-life. If they terminate in galactose or high mannose, then you can decrease the half-life. They can be immunogenic. You heard just now about plant-derived glycoproteins and proteins that carry N-glycolyl acid. They can be highly immunogenic in mammals. So these are all very good reasons why you need to understand your sugars and satisfy the FDA requirements or the EMA requirements for drug safety and efficacy. So nowadays they're asking you to define the carbohydrate content in detail, in a lot of detail now. You have to show the antennary profile, the glycosylation sites, and the site occupancy also. So you need to be able to define mannosylation, galactosylation, fucosylation, and sialation. So in other words, this is really asking for a pretty full glycan analysis of your product. Now, glycosylation is really important for both innovator and follow-on biologics and biosimilars, because you can see from here that many of these patents are now expiring 2016, some of them 2017. So many companies now are positioned to be able to bring on their biologics and biosimilars as soon as these patents run out. And many of these molecules are IgG, but also many of them are erythrocyte stimulating agents, so erythropoietin type molecules. And so you can see here some of them have expired already. So erythropoietin is a very good model of biotherapeutic because, first of all, it demonstrates a number of really important fundamental principles of glycosylation. First of all, glycosylation is cell type specific. So you take one glycoprotein, you express it in one cell, you get a certain type of glycosylation, like we saw in the insect cells, or you express it in CHO cells, and you see different glycosylation. So first of all, you have to choose which cell are you going to express your glycoprotein in. And then the glycosylation is also site-specific. So if you don't have a sugar at this site, the glycosylation at the other sites will be affected by that, and they will be different. So having all the sites occupied is really important to consistency of the types of sugars that you get on your molecule. And also, the glycosylation profile at each site will be different because imagine all those enzymes, they're going to process these sugars. And the efficiency with which they do that depends how exposed these sugars are on the protein surface. Because if they're blocked by the 3D structure of the protein, then you'll get less processing. So every site has its own set of sugars. And also these glycans are really important in erythropoietin because they can extend the half-life. By being fully sialylated, you can extend the half-life. They also stabilize and protect the protein. They can modulate its activity, express them incorrectly in the wrong cells, you'll have antigenic epitopes. And you also might introduce something like a sugar that could take this into the immune system by binding to the mannose binding lectin or influencing C1q binding. So understanding where your glycoprotein is going to go in the patient is now really an important area to begin to understand. So just to show you how important sialic acid can be on erythropoietin, if you have your sugars terminating in sialic acid, it will be in the patient for 3 hours, where it really is effective. If you don't have sialic acid, it will be cleared in 2 minutes, so it does nothing. Just adds to the burden of the patient having to clear all this material. So everyone wants to get their erythropoietin as fully sialated as they possibly can. And it's not just erythropoietin that's affected by this. All these other molecules, you can see if you look at this column, see cholinesterase 17 hours if it's sialated, 26 minutes if it isn't. Even something down here like luteinizing hormone, 1 hour if it's sialated, 9 minutes if it isn't. So keeping your proteins sialated, not letting them be removed by breakdown of cells at the end of the process, for example, is really important. So let's talk a bit more about immunoglobulin, because I guess this is where most people begin with their biosimilars. So this is a schematic of IgG, has heavy chains, 2 heavy chains, each of which has a conserved N-glycosylation site on it. So IgG has 2 conserved sugars here. It has 2 light chains, and sometimes the light chains will carry sugars, but only cetuximab is the only one which carries sugars in the light chains in therapeutics. They're just deleted out. Then the antigen binding sites are here, so these are the parts of the protein which recognize the target. This is known as the antigen binding part of the molecule, and this is known as the FC region where the function happens. So once these tips of the VH and VL regions recognize the target, then something happens down here and you get some function. So different plasma cells in people produce differently glycosylated IgGs. So we have a whole range of different sugars here and also a whole range of different structures in the FAB. But in a monoclonal antibody, of course, these are all the same. They're monoclonals. But in plasma, you have polyclonal IgG. All IgGs have regions here where they can bind to Fc gamma receptors. So Fc gamma are 1, 2, 3, and so on, and also to C1q, part of the complement pathway. And then down here is the region where the IgG binds to the Fc gamma Rn receptor, and also to protein A, protein G, which most people still use as downstream processing to isolate IgG. So you can see these 2 regions are not the same in the same place at all. If you take all the sugars off IgG, you get huge changes. So you pretty much destroy any Fc gamma activity, because if you don't have these sugars here, all this hinge region is destabilized and none of these molecules bind effectively. If you take the sugars off, you can still recover your IgG with protein A or protein G, and FcRn still binds because this region is not really affected if you take the sugars away. But so the sugars are really important for preventing aggregation. But I just put this in because it's not just the sugars that cause problems. The protein itself can be damaged at any stage during manufacturing, and protein damage can lead to aggregation. So protein synthesis and secretion, the bioreactor conditions, the protein purification conditions, the inactivation of viruses, which is often done by heat, the presence of low pH detergents and high salt, and formulation with the buffers, the excipients, and the containers that you use. All of these things can affect the structure of the protein, and that can alter the efficacy and safety of a drug. So if you take if you take Avastin, hydrogen deuterium exchange experiments have shown the changes after aggregation. So if these hotspots in this monoclonal antibody become exposed and you start to get aggregation, then all these surfaces covered in red actually become exposed on the surface of the IgG, and the sections marked in blue, then they all become decreased, they involute into the protein. So the understanding the structure of the protein and how important it is to make sure the protein doesn't become denatured is just as important as understanding the structure of the sugars and making sure they don't assist denaturation or aggregation. And so we looked at a whole series of different monoclonal antibodies, including Avastin, and we were looking to see the effects of cell protein and processing on these different monoclonal antibodies. So this antibody it's recombinant human IgG, and it's CHO cell, and it's expressed in a nutrient medium which has gentamicin in it. We looked at cetuximab, and this is a chimeric IgG1, and it's produced in a murine cell line. And infliximab is a chimeric human murine IgG1 antibody. It has murine B regions, and it's made in hybridoma cells. This is another IgG, rituximab. It's a chimeric mouse-human monoclonal antibody, and it has here human IgG1 C regions, mouse light chain regions, and so on. And then Herceptin is a humanized IgG. It's made in CHO cells, and it's made in a suspension culture. So the message of this slide is that all these IgGs are different in their FAB region because they all have different targets. They differ in the cell lines that they're made in the way that the antibody is put together, whether it's a chimera with mouse or it's a humanized IgG. They're all monoclonal antibodies, but they're all very different from each other, and the type of cell processing is also different for each one. So when you look at the glycosylation, it's not really surprising that you can see there are massive differences. So these are different types of glycans attached to these monoclonal antibodies. So just eyeballing this, you can see they're all very different from each other. So human serum IgG has 4 subclasses, 1, 2, 3, and 4. And most of the therapeutics on the market at the moment are of the IgG1 subtype. There are some others, but most of them are IgG1. And these are some of the targets for FDA-approved monoclonal antibodies. So some of them are just blockers, they're inhibitors. Others are focused towards autoimmune disorders, and these are their targets. TNF-alpha is probably one that you've heard of a lot. These are anti-cancer antibodies. These are for other purposes. This is for allergic reactions. It focuses on IgE. And tissue rejection, of course, another important use of monoclonal antibodies. This is a model we just recently made of Campath, that's anti-CD52 antibody, and it's really, this is CD52 with a GPI type anchor, a very small peptide, and a massive sugar attached to it. And the antibody actually attaches here in this tiny space. So of these FDA-approved monoclonal antibodies, most of them are IgGs, some are just Fab regions, some are single-chain FBs, but they're mostly IgG1, IgG2, IgG1 fusion proteins, IgG4, and so on, IgM and IgY. And so you heard about this earlier, but I'll say it again, that the critical quality attributes for glycans are really important because this is what leads to safe drugs that actually work, and also if you're going to get your drug past the regulators, you have to be able to demonstrate that you either do or don't have these terminal residues depending on what the function of your IgG is. So if you want your IgG to interact with the mannose binding lectin to activate complement, then as you saw before, you have to have N-acetylglucosamine terminating these sugars, because that's the only terminal sugar on IgG that will activate mannose binding lectin. If the sugars terminate in galactose, you can get placental transport, and you can also get increase in C1q activation. If you desilate your IgG, you'll lose its anti-inflammatory properties, because silated IgG is anti-inflammatory. If you don't have core fucose, then you will enhance antigen-dependent cellular cytotoxicity, which you might want to do. So you have to know the function of your monoclonal antibody, because you may or may not want these terminal residues. And there's also this problem of antigenic activity. So 50% of us could have antibodies against these plant epitopes. Then we all have antibodies against N-glycosyl-neuraminic acid, and that might prove to be a problem in some people if you give them antibodies with, if you give them monoclonals with N-glycosyl-neuraminic acid on. And then alpha-galactose, so this galactose residue linked in an alpha position to another galactose, that's not something that we can put on. We don't have the right enzymes to attach this structure, and we're highly allergic to it. It's present in pigs, and the very first pig valves that were made were rejected, and people died of anaphylactic shock because they had this alpha-gal residue on, and people didn't realise how serious that would be. Sugar is also really important for clearance. So as I showed you with the EPO, it can, the length of time your drug is in the patient is clearly really related to efficacy. So if you have mannose structures, you get rapid clearance. The fucose makes no difference to clearance. GE0 has a longer half-life for a particular IgG subtype. Terminal N-acetylglucosamine can also be rapidly cleared, and agalactosyl IgG is also rapidly cleared. So understanding again your PK, your pharmacokinetics in the patient is something that's really challenging and involves glycosylation. Now, in terms of functions of these antibodies, some of them are simply blocking. So chicken IgY, it blocks the binding of Pseudomonas bacteria flagella to cells and tissues. So here are the bacteria, here are the flagella, and this is IgY. It just glues onto the flagella, which means that this bacteria then can't bind to cells and tissues. It's not really going to, the antibody FC is not going to do anything, it's just blocking the binding to the cells and preventing its activity. And the same with Humira. This is an anti-TNF antibody, and it's a blocking antibody. It's binding to tumor necrosis factor, and it's just stopping it being active. And you can see here TNF alpha it's important in inflammatory processes, for example. Lots of pathways affected by TNF alpha. So to dampen down the acute phase response, you want to mop it up, and this is what Humira does. But then there are many other antibodies where you actually want some function from the Fc region of the molecule. So here are just some of the receptors that IgG-Fc can bind to. The neonatal receptor, that has some influence on recycling of IgG as well as transport across the placental barrier. Then there are these Fc receptors, so the whole range of Fc gamma receptors here, and some of them initiate ADCC very well, like Herceptin is designed to do that. And Rituxan here will operate through Fc gamma R2. C1q binding to initiate the complement receptors, the complement pathway, to get CDC activity. And then other receptors like DC sign, these other C-type lectins can carry molecules into the immune system. So you need to know whether you want to invoke ADCC or ADCP, phagocytosis, whether you want CDC activity, or whether you want your therapeutic to interact with the immune system. So all the time, it's not enough just to know what sugars you have. You have to understand which sugars you need to get your drug to be effective. And some drugs, these here, for example, really depend on ADCC. You want your monoclonal antibody to bind to targets on the tumour, for example, and then you want their Fc regions to interact with the killer cell so that you can have your tumour cell killed. So all of these drugs are all dependent on having effective antigen-dependent cellular cytotoxicity. So you need them to bind to these Fc receptors. Now, we did a lot of work on Fc receptors and published a review because these receptors are very heavily glycosylated. So all the pale blue here, these are all sugars. So you can see the Fc receptors have huge protection. And of course, the site where they bind to the Fc of the IgG has to remain open in order for the activity to take place. But the Fc receptors have actually different affinities for the different IgG subclasses. So Fc gamma R1a, for example, doesn't bind to IgG2 at all. It binds to IgG1, 3, and 4, but not IgG2. So if you have an IgG2 and you're expecting it to bind to Fc gamma R1a, well, it won't. So understanding your activity of your antibody for these different receptors becomes really critical. So this is for the Fc gamma R3, showing you how the different subclasses bind, and so on. So they all have well understood different specificities for these different Fc receptors. And the whole thing is more complicated than this, and this is a subject of very active research at the moment because you have a range of Fc receptors and they're expressed on all different